This video is Chapter 28 Notes, Part 1, an introduction to the nervous system. The nervous system being your brain, spinal cord, and all of the neurons throughout your body. The nervous system is a way of communication between cells. So again, we will see the idea of a molecule being released by one cell and binding to a receptor on a neighboring cell. One major difference between the nervous system and the endocrine system is that the endocrine system relies on blood. They say it takes between 1 to 2 minutes for one of your blood cells to travel an entire loop through your body. So the endocrine system is a little bit more slow in comparison to the nervous system, whereas if you were to stick your finger on the top of your hot stovetop, it would not take you 1 minute to realize that the stovetop is hot. So the nervous system is much faster because it does not rely on the bloodstream to send a chemical message. To get us started, the main type of cell in the nervous system is called a neuron. Shown here is a diagram of what neurons look like, and there's a few different parts that are labeled that we'll go through. The way that my neuron is oriented is left to right, meaning that it would receive a signal, it would then respond and release a signal towards the right. Of course, your body is three-dimensional, so a neuron could be oriented in any direction. It's just that always the signal and message starts at the dendrites. So the dendrite is where the input is received. That input could be a chemical, meaning it could be a molecule, or it could be a sensation, like something that you see, like a marker flying at your head. The input would be a sensory input. There's no molecule from the marker that's coming at you. It's just you would be able to respond and move to the side because of the sensory visual input. So the dendrite is what receives the input either from a sensation or from a chemical. The cell body allows for normal functioning of the cell. So in the cell body, we have the normal everyday tasks, such as breaking down sugar to make ATP. The cell body is where we would find all the organelles that allow the cell to just function normally and stay alive. The long part of the neuron is the axon, which will be the focus of most of the notes today. The axon is where the action potential travels. We'll spend a lot of these notes today talking about what the action potential is. On that axon, there's myelin. Myelin allows the action potential to travel faster. And then the end of the neuron is called the synapse, or it also goes by the name terminal, and that's where the chemical will be released to communicate with the neighboring neuron. We call that chemical a neurotransmitter. So we're going to go through all of this in more detail here and in the next video. When a neuron receives input, that input has to be on the dendrites, and then the message travels through the neuron and gets released at the synapse. So a message in a neuron can travel only one direction. You would not see a chemical binding to the synapse and moving the opposite way. No. So because of that, neurons have to be oriented in a specific pattern as shown on this slide, where you have the synapse of one neuron ready to release a chemical such that it can bind to the dendrites of the neighboring neuron. In reality, in your cells, there's probably 10,000 different neurons with their dendrites arranged around the base of this synapse. This diagram shows only 2 neurons here, but of course the speed at which neurons communicate in your body is extremely fast and can go in all different directions to send messages up to your brain, down to your toes. Your neurons are communicating in all different directions. So in reality, there's thousands and thousands of neurons oriented around the synapse of a single neuron. However, if I had a synapse aligned to a synapse, they would not be able to communicate. The signal has to be released at the synapse and bind to the dendrite of the neighboring neuron. We're now going to spend the next few minutes talking about the action potential, and then we will come back in the next video to discuss more about what happens in this space between neurons. So now a bit about the axon and the action potential. The axon is the long tube in the neuron. So when I think of my neuron here, these are my dendrites where I would receive the signal. There is a long part of the cell called the axon, and then the terminal at the other end. So we're now going to focus a bit on what's happening in this tube called the axon. To remind you, we are talking about a cell. A neuron is a cell. So around the axon and around the entire cell, there is a phospholipid bilayer. So here I'm using a box to represent a zoomed in part on that axon, but don't lose sight of the fact that this is a cell and thus it is surrounded by a cell membrane. That axon is 3-dimensional. It looks like a flat box in my picture, but in reality it's more like a tube. Like if you imagine a paper towel tube or a toilet paper tube, the roll that those products are on, it's a 3-dimensional tube. So there's an inside and an outside. There is inside the cell and there is outside the cell. The barrier is the cell membrane. The cell membrane, to remind you, allows some molecules to squeeze through on their own, but other molecules and ions require a channel due to that inner hydrophobic region. So the phospholipid bilayer is made of hydrophobic tails that will keep charged particles from just slipping through the membrane. It will also keep large particles from slipping through the membrane. So the cell membrane acts as our barrier between the inside and the outside of the cell. All right, so when we are talking about the axon, we can talk about a neuron either firing or being at rest. I have separated my image here into 4 slots, and I will go through each of those 4. In slot number 1, I am going to show the neuron at rest. When a neuron is at rest, it has a separation of charge. So when your neuron fires, it is a movement of charges. The 2 main ions that the nervous system uses are sodium and potassium. When a neuron is at rest, there is a huge accumulation of sodium ions on the outside of the cell. The cell spends energy to put them all on the outside. More on that in a moment. The inside of the cell contains potassium ions in a greater concentration than the amount of potassium found on the outside. So there's a huge amount of sodium on the outside and a little bit amount of potassium on the inside. In biology, we call this a separation of charge, and we would say that the inside of the cell is less positive, and when we talk about neurons, we call that negative. So just be a little bit aware that it's kind of weird. It's negative compared to the outside, but in reality, it's just less positive. I would call the outside of the cell positive due to all of that sodium. So when my neuron is at rest, it's an accumulation of positive charges on the outside of the cell. If we stick an electrode, a little pin inside of a neuron, we would be able to measure the charge difference between the inside and the outside of the cell. And when the cell is at rest, it is 70 millivolts less positive than the outside. So I would say that the neuron has a charge of negative 70 millivolts. That's kind of all outlined up here on the top. It says that when the cell is at rest, we also call it polarized. Polarized means opposite, like the North Pole and the South Pole are on opposite sides. So we call it opposite because one is positive and one is negative. And that is when the neuron is not firing. During that time, the outside of the cell is positive and the inside we call negative. There is a pump that the cell uses to push the sodiums to the outside. We call that the sodium-potassium pump, simply because it moves sodium and potassium ions. The neuron purposefully sets up this separation of charge using energy, using ATP. The cell is going to pump all the sodiums out. It is going to allow potassiums to come in, and that creates then our separation of charge, where outside of the cell is positive and inside the cell we call negative. After the neuron has set up that separation of charge, it could then send a signal. That signal is what we call the action potential. When the action potential sends, we call that depolarization, meaning the separation no longer exists. So what happens is that our cell was at rest, as shown in image 1 is at rest. At image 2, what we have occur is all of the sodium then rushes into the cell. It's kind of like opening the floodgates. Suddenly, all the sodium opens. There are gates in this phospholipid bilayer that open, allowing the sodium to come in. When the sodium comes in, a little bit of sneaky potassium drifts out. Because that membrane is just so open that the cell kind of loses track of what's coming in and out. It says that up at the top. So sodium then rushes into the neuron. It is controlled by gates. So the cell will choose when to open those gates. That's what's coming next. And when those gates open, sneaky potassium drifts out. When that happens, and for that split millisecond, when all of that sodium has rushed into the cell, we have now created a huge positive charge inside of the cell because all that sodium came in. So it then has a charge of about positive 50 millivolts due to the movement of sodium into the cell. A little bit more about the gates. The gates are in the cell membrane. So we see that here's my phospholipid bilayer. Charged particles cannot pass through those hydrophobic tails, so they require protein channels. Generally, those protein channels are closed, keeping sodium on the outside of the cell. However, when the cell receives a signal to open the channel, sodium can then go through. It's a little bit, again, like a domino effect. Once one sodium channel opens, it causes a shape change in the neighboring sodium channel because, as we recall, channels are made of proteins. Proteins fold very specifically where positives and negatives pair together. Hydrophobic regions point in, hydrophilic regions point out. So I can change the shape of a protein by changing the charge of the environment it is in. So when this channel opens here and sodium goes through, the charge in this green area changes, causing the next protein channel to open because of a shape change. So that is what I mean by they sequentially open. When one opens, its neighbor opens, its neighbor opens, its neighbor opens. It's a domino effect that sends an opening sequence of channels down the length of the axon. Then, after those sodium channels have opened and we have a positive charge inside the neuron, we are then ready for the neuron to recover. So period 3 is called refractory or recovery. If the cell did not recover, it would never be able to send another message again because the message is sent by a flip-flop of charge where sodium rushes in, potassium sneaks out, and that travels the length of the neuron. So when the neuron receives a signal, the first few sodium channels open, and it's a domino line down the axon until the last few sodium channels open. After the sodium channels open and we have all of the sodium inside of the cell needs to recover so that it could send another message in the future. So the way that it recovers is by pumping that sodium back out and allowing the potassium to come back in. So that is refractory, aka recovery. Again, we have the pump that is using energy to return the cell back to rest. That will then allow another action potential to come. That's what's shown on the following slide. Period 4, we are then back to rest. When we are at rest, again, we have all the sodiums on the outside and a few potassiums on the inside. So we have returned back to how the cell was originally. So this is at rest. This is also at rest. My signal has traveled from left to right. So you might be wondering, why did I seem to go in the opposite order where 1 is on the right and 4 is on the left? And that's because if I took a snapshot of a neuron and my signal is traveling left to right, if I took a photograph, here on the left would already be completely recovered. 3 would be in the process of recovering, 2 would be the action potential, and 1 would be waiting for the action potential. It's due to the orientation of the way that the picture is. Myelin is a substance that wraps around the neuron. It wraps around the axon, and it just allows the signal to go a bit faster. The way that it works is that a sodium channel before the myelin and a sodium channel after the myelin can cause each other to open and close as opposed to waiting for every single sodium channel in between. So myelin is said to insulate the cell, allowing the action potential to travel faster, simply because not as many sodium channels need to open in order to send the action potential. So to conclude, you will see some action potential graphs throughout the nervous system unit. And what we measure on the action potential graph is the voltage in the neuron. So we are measuring the amount of charge inside the neuron. To remind you, when the neuron is at rest, there's a huge amount of sodium outside of the cell. That causes the neuron to be very negatively charged. Then some sort of stimulus happens. Sensory input or a chemical input causes some of those sodiums to start to go into the cell. We call that the all or none point, meaning your body does not respond to every tiny stimulus it gets. Imagine if you were sitting at your desk and you felt every particle of dust hitting the top of your head. You would be going completely crazy. Or if you heard every single person in the room inhaling and exhaling, it would drive you crazy. So we call the all or none point the stimulus that causes a neuron to fire. Once the neuron has reached enough stimulus input, it will then send the action potential. The action potential is simply when all of the positive sodium ions rush into the cell. So the action potential is not a molecule swimming through the length of the axon. No, there is no molecule called an action potential. All the action potential is the brief microsecond where sodium moves into the axon and back out. That's it. So when that sodium ion is inside of the axon, we then see a positive charge. We call that then the action potential. After the action potential has been sent, the neuron needs to recover. If not, every neuron would only be used one time and never be able to be used again. So the neuron will then use the sodium-potassium pump to pump those sodiums back out. And as those sodiums are being pumped back out, the voltage is dropping because the positives are leaving the cell. Sometimes the cell makes a little bit of an error and pumps too much sodium out, and then it goes back to rest. So at the beginning and end of my diagram, the voltage is the same. The neuron is at rest, ready to fire. This would then continue to go on repeatedly. So my neurons would continue to fire until the response was given or the stimulus stopped. So that's shown detailed for you on the next image. The cell is at rest, voltage is negative. There is an all or none point where the neuron either will fire or not. If the neuron fires, it becomes positively charged due to the sodium rushing in. After that, the neuron needs to recover. Sometimes it needs to fix itself for a brief microsecond, and then the neuron is back at rest. We still have not yet talked about how the signal causes the neuron to fire or how the signal gets released at the end of the synapse. So more on that in video number 2. But big picture from this video, you should know the parts of the neuron, what is an action potential, and how is the action potential controlled by a flip-flop of charge, and then also how does voltage change when the charges move position.